So we now move to Jean-Léon Maître from Institut Curie, and he will be talking about hydraulic fracturing and coarsening position, the lumen in the mouse blastocyst.
All right, thank you very much, Thomas. So I'd like to tell you about what we do in the lab, which is mostly about the early development of mammals. We mostly work with the early mouse embryo, but we also start working with human embryos. And what I would like to tell you about today is the first story that we are trying to report from the lab, which is about hydraulic fracturing and active coarsening and how this is positioning the first lumen of the mouse embryo. So during the very early phase of mammalian development, the embryo is undergoing what is called pre-implantation development, which is before the implantation of the embryo into the uterus. And so during this time, we can culture and observe the embryo developing in vitro. And so this is a mouse embryo under a microscope, which is undergoing cleavage divisions, which are divisions without cell growth during the interphase. And then at the 8-cell stage, the embryo will start to change its shape to build this very characteristic architecture, which is called the blastocyst. The blastocyst is a structure here which is composed of two distinct cell types. There's one epithelial layer, which is a flat squamous epithelium, which is forming an envelope. And the job of this epithelium, which is called the trophectoderm, is to implant the embryo into the uterus and to contribute to some of the tissues of the placenta, and it will not contribute to the embryo proper. Inside this envelope, there is a fluid-filled cavity, which is called the blastocell, and there's a cluster of cells here, which is called the inner cell mass, and this inner cell mass will make all of the cells of the mammalian body. What we try to understand in the lab is how you can build this specific architecture. And you can break it down in 3 different steps, which conveniently in the mouse happens at the 8, 16, and 32 cell stage. The first one is called compaction. I will briefly tell you about this. Then the formation of the inner cell mass needs cells to be internalized inside the embryo, and then there's the formation of this lumen. So very briefly, what we do in the lab, we try to characterize the mechanics of the embryo. We try to characterize the shaping of the embryo by using microscopy, biophysical tools. And one of our favorite methods is a non-invasive way to measure surface tension. And here, this embryo at the 8-cell stage, it is undergoing compaction. At the same time, we are measuring, mapping out all of the tension of all of the interfaces of this embryo. And what we could see is that during the 8-cell stage, the embryo is compacting because cells are increasing the surface tension, specifically at the surface of the embryo, pulling the cells together. And this is due to an increase in the contractility of the actomyosin cell skeleton, which I do not need to explain to you by now, I think. So we can also then use those information to discuss with theoretical physicists. Here is a simulation from a model from Hervé Turlier, who is in the room and in Collège de France with his lab at the moment. And we can also then recapitulate what we observe in the experiments. Afterwards, at the 16-cell stage, what we see is that some of the cells will have different surface tensions, and the one with the highest surface tension will become internalized. This is due to an asymmetric division of a contractile domain, which is given to some of the cells only. And so because we know this, we can now manipulate the contractility of the cells to actually direct cells towards the inner position. And here is an experiment where we have built a chimeric embryo with a cell here which has a strong surface tension and a host which has a weak surface tension, and this cell here is invading this embryo and becomes part of the inner cell mass. And again, we can faithfully recapitulate this process in the simulation that Hervé can do. Now, what I would like to tell you is about a very different process. This was about building cell-cell contact, rearranging cell-cell contact. The formation of a lumen requires the contacts to be separated. And here is a process of this lumen formation. The blastocell forms between this trophectoderm layer and the inner cell mass cell cluster. And so you need now to basically separate those cells. So what do we know about lumen formation in general? Lumen formation has been studied in vitro mostly in the context of the formation of apical lumens. What happens typically is that cells will bring the apical membrane together to form some kind of rosette-like structure. And what's important here is that this is a polarized epithelium. And what you want is you want to polarize many different things inside the cell. One of the important components which needs to be polarized is the transport of osmolytes, like ions, like sodium and also protons. So you want to pump this osmolytes into the apical compartment, and then this will draw water from the outside of the cell cluster, and the water will just be pumped inside through aquaporins. You want then to keep everything inside this compartment, and you want to make this compartment tight by having tight junctions, which are here in green. And also what you want, ideally, is this surface here, which needs to separate. You want it to be as little adhesive as possible. You don't want it to be sticky. And so what the cells are doing as well is that they are removing all of the adhesion molecule from this interface. All of the cell-cell adhesion molecule are on the lateral side, and the basal side contains the integrins which are binding to the extracellular matrix. So this is a weakly adhesive interface. Sometimes you even have proteins like polycolixin, which can be localized at this apical membrane, which will actually serve as a repellent. They are strongly negatively charged proteins which will actually help the membrane to detach. So once you start to pump some water in there, it's going to be very easy to separate those membranes. In the mouse embryo, we have a basolateral lumen. The polarity is inverted, and the lumen forms on the basolateral side of the cell, which contains all of the adhesion molecule. The rest is fairly similar. So the tight junction are at the surface of the embryo. They keep the fluid inside. The transport of osmolyte is still directed towards the intercellular space within the tissue, but we still don't really understand how you can form a basolateral lumen. And for this, we have very little information. So we would like to understand how you can form a basolateral lumen in the context of the formation of the blastocyst. And the other question we would like to answer is, how do you position this lumen? And for the mouse embryo, it's very important. The embryo initially is radially symmetric. It has no axis of symmetry. But at the end of the formation of the lumen formation, we have now the first axis of symmetry of the embryo, with on one side the inner cell mass, and on the other side this lumen. And this is very important for the development of the mammalian embryo, as it will help directing the dorsoventral axis of the embryo later on. So when you form an apical lumen, typically you form a rosette, and so you bring the apical sides together, and then the lumen forms from this seed, which just expands. So do we have here a local pumping or local disruption of cell-cell contact, which will then just expand and form the lumen there? So to check that, what we did was to look into the formation of this lumen under a microscope with high temporal and spatial resolution. And here is a 32-cell stage embryo, which is about to start the formation of the lumen. And what you see is that all cell-cell contacts are forming some small bubble-like structure, which we call microlumens, which transiently appear and then disappear throughout the embryo. And basically, all of those microlumens start to grow, and then they shrink, all but one, which becomes the blastocell. But all cell-cell contacts are transiently broken, all of them, including the contacts between the cells of the inner cell mass, which you don't want to separate. You want the inner cell mass to stay nicely cohesive. So we have analyzed this phenomenon, and so what we see is that we have typically two phases for those microlumens before they start to form the blastocell. So we distinguish two types of microlumens, the one which are between two cells, right, the red ones here, and then the one which are at the corners between multiple cells. And basically, they both show the same kind of dynamics. So they grow, and then they shrink. They grow, and then they shrink. It's just that the scale at which the multicellular microlumens grow and shrink is much, much faster, about an order of magnitude faster. But it's the same kind of dynamics. What we see as well is that the multicellular microlumens tend to peak about half an hour later on average than the bicellular microlumens. But we have this characteristic growth and shrinkage phase. So we try now to understand what drives this growth and shrinkage phase, which we call sometimes the swelling and discharge phase, for reasons which you will soon understand. And at first, we wanted to get some hints about what could be happening during this separation. And so, as I said, there's very little information about the formation of basolateral lumens. But there's one paper which was key for us to understand what is happening, which is a very, very nice paper from the lab of Xavier Trepat. What they have done is that they have taken epithelial cells, they have put them onto a hydrogel, a sponge-like material, loaded the gel, and then expelled all of the fluid through the basolateral side of this epithelial monolayer. And this basically just breaks apart the cell-cell contact. And what they see then is the formation of transient structure, which are about this shape. The adhesion molecule are relocalized to the side, and basically you just transiently just flush some fluid through the monolayer. The contacts form this little bubble structure, and then they just heal back. And so this is hydraulic fracturing, or fracking. You basically just flush some fluid through the cell-cell contact and just breaks them. And so we thought that those structures look very similar, and so we investigated whether there could be hydraulic fracturing happening in the embryo. So as I mentioned, during this process here of hydraulic fracturing in MDCK cells in vitro, the adhesion molecules, ecadherin, are relocalized to the side when the fluid is pushing through. And so we looked into the localization of E-cadherin using E-cadherin GFP transgenic embryos. This is a membrane here, which is in magenta here, and in green is the E-cadherin GFP. And so you see that when the bubbles are forming, the E-cadherin is being relocalized. It is initially rather homogeneous at the cell-cell contact, and then it becomes more patchy. You can see that better maybe on those still images. This is before the formation of the microlumens. E-cadherin is already a bit patchy, but it is more homogeneous than later on. When the microlumens are forming, you can see that E-cadherin is enriched at the sides of the microlumens. We are measuring the heterogeneity level of E-cadherin along the contact, and we see that it's increasing. That's just the metrics that we use to follow this process. And then we thought, okay, E-cadherin is being relocalized. Is this relocalization depending on the accumulation of fluid? So we blocked the accumulation of fluid. We did that in multiple ways. One is to prevent the correct apicobasal polarization of this embryo. It will not be able to pump in a directed way the osmolytes, and it will not form a lumen. Or we can also override completely the osmotic gradients that the embryo can form by dumping sucrose into the medium. So this is a control embryo. It forms microlumens and then a blastocell. E-cadherin is relocalized. In this case here, polarity is affected. It is not able to, the embryo is not able to direct the pumping of the fluid, and E-cadherin remains more homogeneous. In this case here, with the sucrose, the embryo again cannot pump water in the intercellular space, and E-cadherin remains more homogeneous. We can measure this again, and what we see is that in the control, the signal becomes more heterogeneous. It is not the case when there is no fluid accumulation. So for E-cadherin to be relocalized, you need the accumulation of fluid, which is compatible with this idea of hydraulic fracturing. So if fluid is coming in and basically breaking the cell-cell contact, what you can imagine as well is that the amount of adhesion that you have at a certain interface will also influence the fluid flow and where it can go. So if you were to pattern cell-cell adhesion, one side more adhesive, one side less adhesive, you would imagine that the fluid would go towards the side which is less adhesive, the path of least resistance. And so we decided to pattern cell-cell adhesion in our embryos. For this, we did the following experiment. We built chimeric embryos made out of either wild-type embryos or a maternal E-cadherin knockout embryo, which lacks a maternal allele of E-cadherin. Those embryos are viable. They are fine. They make an embryo. They make a blastocyst. They are viable, but they have less adhesion. And so when we build those chimeric embryos made out of embryos which have the normal level of E-cadherin or a lower level of E-cadherin, what we would expect is that the fluid would go towards the side which has less adhesion. So this is a wild-type chimeric embryo, and we see that the cells are composing all kinds of tissues. The lumen is in any position at the periphery of the embryo. And here, if you look at the distribution of the cells in the three different parts of the embryo, we have the inner cell mass, and then we have two kinds of trophectoderms. One which is called the mural trophectoderm, which is lining the blastocell, and one which is the polar trophectoderm, which lines the inner cell mass. And so when we look at the maternal E-cadherin knockout wild-type chimeric embryos, the lumen forms almost systematically onto the side of the E-cadherin knockout, and the trophectoderm that is formed by the E-cadherin knockout cells is almost exclusively the mural trophectoderm, the one which is lining the blastocell. So this is compatible with the idea that the fluid would come and be basically the fluid flow would be resisted by adhesion, and this is compatible with the idea of hydraulic fracturing. What we can also say is that patterning cell adhesion is sufficient to position the lumen, and therefore it is sufficient to position the first axis of symmetry of the embryo. So the first question was, what controls the position of the blast cell? Well, patterned cell adhesion can control the position of the blast cell and can orient the first axis of symmetry of the embryo. So this is one way that we could imagine of how the embryo could position its axis. So we looked into any kind of pattern of adhesion. One way to look into that is to look at this embryo, which is undergoing hydraulic fracturing, and we see that, for example, this side here has a bit more cracks than this side here, but eventually the blastocell forms on the other side. And we looked in more detail into the patterns of fracturing, the rate of formation of those lumens, the concentration of E-cadherin, the heterogeneity of E-cadherin, and we could not find an obvious pattern of adhesion or fracturing which would explain where the blast cell forms. So we looked into the second phase of the formation of those microlumens, which is what we call the discharge phase. And so how do you basically then make those bubbles appear everywhere and then resolve this global phenomenon into just one position. And so when you look at even higher temporal resolution at this phenomenon of microlumen formation, so here this is a movie on which we are looking at those microlumens every 5 seconds on a stack using light sheet microscopy. And so here we have a fairly large microlumen in the center. We also have smaller microlumens, which you will see once the movie will start on the neighboring junctions. And what you can see is that this microlumen is increasing in size on the time course of about 10 minutes. If you look with sufficient time resolution, you can actually see that there are pulses of growth of this microlumen in the center. What you can see as well is that this microlumen here is eventually merging with this one. There's some merging of the microlumens, but most of the microlumens which are in those junctions, which I hope you can see, they do not seem to flow or coalesce together with the growing microlumen. It doesn't look like there's much flow of the microlumens themselves. Instead, what seems to be happening is that the microlumens are growing or shrinking. So what could be happening is a process which would be analogous to what's called Ostwald ripening, where we have two different droplets or soap bubbles which have different sizes. And for example, here in an emulsion of oil and water, it's the same surface tension. Because of the Laplace law, if two droplets have different sizes and the same tension, the small one will have a smaller pressure than the larger one, and this pressure gradient will cause the small one to shrink and the large one to increase in size. And eventually, you will just form one big droplet. That's what you see when you do your vinaigrette. So here what we have is a situation which is a bit different. We don't have a diffusion of material from this droplet to this other droplet. We have microlumens, little bubbles, which are connected by this intercellular space. And so the idea would be similar. If you have a pressure gradient, you will get fluid flow from the small microlumen to the larger one if you have this homogeneous surface tension and different radius of curvature. So to see whether there is fluid flow between microlumens, what we have done is we have bathed our embryos in a photoactivatable dextran, which is trapped between the intercellular space. And when we photoactivate this dextran in this large microlumen, you can see those little pulses of photoactivation that we do over the time course of about a minute and a half. What you can then check is whether the dextran can be found in the neighboring microlumens. And so this is the quantification which is there. Basically, what you see is that this is the pulse in the microlumen that we photoactivate. It first peaks, and then you have a decrease in signal, which could correspond to the dextran diffusing away from the photoactivation zone. And then you can find some signal in the microlumens which are further away, which indicates that the dextran can travel from one microlumen to another within short time scale. So the idea would be that the liquid could just travel within this intercellular space. So now if you can allow the fluid to flow from one microlumen to another, you can then direct the fluid flow by different means. One is the classical Ostwald ripening way. If you have an homogeneous surface tension in this material, the small one will have a lower radius of curvature and therefore a higher pressure and empty itself into the larger one. But what you can also do, because cells and the embryos are not passive materials, which is where you usually find Ostwald ripening, they can actively control this other parameter, which is the surface tension. And so now if you have two different microlumens which are connected, this one is smaller than this one, but because it has a lower surface tension than the blue one, now the flow is reversed, and it is the smaller one which is gaining all of the fluid because it is less tense. So this is the case for two connected microlumens. The embryo is more complex. It has many microlumens which form a network, and this network will also influence where the fluid will flow. The connectivity of the microlumen will be very important. If you're in the center of the embryo and you have many microlumens from which you can draw water, or if you're at the periphery where you have very few microlumens which constitute a reservoir of liquid from which you can draw, then the dynamics and the final outcome of where the lumen will go will be very different. So again, this is, I didn't mention that, but this is the work done in the lab of Hervé Turlier. These are simulations where we have built artificial networks of microlumens which are connected. And then if the tension is homogeneous, because these guys here are more connected than the one at the periphery, the lumen will form at the center of the embryo, in the ICM, in the inner cell mass, which we don't see in the embryo. In the embryo, what we see is that the lumen forms at the periphery. But now if we pattern the surface tension and make it so that the surface cells have a lower surface tension than the cell of the inner cell mass, then the lumen can form at the periphery of the embryo, which is what we observe in our experiments. This difference in surface tension we know already exists. We have measured that previously. The reason the inner cell mass cells go inside is because they have a higher surface tension, because of this higher contractility. Nevertheless, we checked that we could see some signs of differences in surface tension just before the formation of the blastocell. And here what you can see is an embryo with inner cell mass cells and trophectoderm cells. And you can see that the interface between the ICM and the T is curved, with the ICM bulging into the trophectoderm, which is indicative that there's either or both a pressure or tension difference between the two cell types. If you reduce contractility, which is responsible for generating the surface tension using a drug called blebistatin, then the interfaces become flatter. So there are differences in surface tension between the cells. They are probably due to differences in contractility, and they are at the level of the cell-cell contact. You can also find those differences in curvature if you look more closely into the microlumens. The microlumens are most of the time symmetric with, for example, those guys here. And if you look at the trophectoderm interface, most of the microlumens are symmetric. If you measure the radius of curvature on either side, you find the same. But if you look at the trophectoderm ICM interface, often those microlumens will be asymmetric, and almost systematically, in more than 80% of the cases, the microlumen will bulge into the trophectoderm. So those microlumens, they are under pressure, they push in all directions, but they deform more the trophectoderm, which is again indicative that this trophectoderm cell is either under lower pressure or lower surface tension, or both. So now the idea is that we have those two different tissues with a lower surface tension in the trophectoderm, a higher tension in the inner cell mass, and this leads the lumen to form at the periphery. If we were to pattern inside the trophectoderm the surface tension, we could direct the positioning of the lumen. And so this is a simulation which recapitulates the experiment which we are going to do, which is to pattern surface tension by using chimeric embryos, now made out of wild type, and maternal myosin knockout embryos. Those maternal myosin knockout embryos are viable. They form a blastocell, they form a blastocyst, they form a mouse, they are fertile, they are fine. They have a surface tension which is 3 times lower than the one of the wild type. And when we build those chimeric embryos made out of high tension and low tension, the lumen is forming on the side of lower tension with the mural trophectoderm, which is lining the blastocell, primarily composed of maternal myosin knockout cells. So just like for the Echidrin knockout chimeric embryos, we can direct the position of the blastocell, we can direct the position of the axis of symmetry of the embryo by patterning contractility and surface tension. So I've told you 2 different ways now to direct the positioning of this blastocell, direct the positioning of the first axis of symmetry of the embryo. Because we see no obvious pattern of adhesion, but we do have a clear pattern of contractility within the embryo, what we think is happening is that what controls the position of the blastocell is a pattern of contractility. So what we propose now is that we have a contractility-mediated coarsening of those microlumens, an active coarsening of those microlumens, which is formed, those microlumens being formed by hydraulic fracturing of cell-cell contacts, which is influenced by the amount of adhesion that you have. And together, this positions the first axis of symmetry of the mouse embryo. This is a lab. We are here in Paris, in Institut Curie, and this is a project which was led by a postdoc, Julien Dumortier, in the lab. He got a lot of help from 2 engineers in the lab, Francesca Tortorelli and Ludmila Deplater. This is a collaborative work together with the lab of Hervé Turlier, who is a theoretical physicist, and 2 PhD students contributed to making the series and simulations, Mathieu Levercher-Andour and Annette Milke. We get a lot of support from Institut Curie, who has excellent facilities, and I want to thank all of them and you for your attention.